Welcome. In this video slash podcast, I'm going to be discussing microbes in sourdough. This is related to my blog, the Sourdough Chronicles, that has been recording the journey through this experiment. If you'd like to see all the details, check out the blog link in the description. Essentially, this all started with sourdough starters. Now you may be thinking, what's that? Bread? To which I say, kind of. It's the start of the typical bread everyone thinks of. By taking some flour and water and regularly replenishing the mixture, the necessary microbes needed to make the bread grow. The yeast that finds its way into the starter feeds off the water and elements of the flour and grows. As the yeast eats, the entire starter will rise and fall with the expulsion of various gases from the reactive process. Over time, the mixture begins to grow more dough-like until it is matured and ready to be baked into bread. So North Carolina State University has been working on what is known as the Sourdough Project for many years. Specifically, they have been researching how the microbes contained in sourdough starters differ by what the contents are by adding a variety of fruits to the usual flour-water mixture, and then observing the results. In the genetics course I participated in, members of the class created a control or normal starter and 1 to 2 others that had a different fruit in it. For my samples, I had my control and also had a sample with a raspberry in it. Then for a series of weeks, I would regularly feed the sample and record its phenotypic characteristics. Eventually matured, samples were sent back to the lab at NC State, where the starters went through DNA sequencing. As the starters were growing, the substances added to the samples, as well as the environment, contributed an assortment of microbes that then grew and cultivated in the starters. By DNA sequencing the contents, we are able to identify what types of microbes are in the starters. In the actual process of sequencing the DNA, the DNA is extracted from the sample and then sent and replicated. The DNA is then sent through a process where a computer will read and organize the base sequence that make up DNA. Once the sequence of different strands of DNA are known, they can be matched to already known samples to identify what type of fungi or bacteria or other organism it might be. In this instance, the results were mostly bacterial with some fungal. So, having access to this information of our own samples as well as mine, specifically and the group as a whole, we were tasked with creating a research question to pursue with the available data. Considering all the time I spent with my raspberry starter, I decided to create a question relating to that subject. As such, the question that I created was, do all berries result in similar microbial compositions? So, through this, we'll be focusing on berries. In selecting which samples were available, which samples I was going to analyze, I did some research into the classification of a berry. In addition to your standard strawberry, raspberry, blueberry, which are all samples I used, I also found bananas, kiwis, and figs, as well as grapes, are considered berries, and they were analyzed as well. In total, I had 18 samples to compare of the microbial composition. So after collecting all the samples I was going to use and running them through software to compare them to one another, I was given these results. So pictured here are 2 graphs. On the left is the fungal composition of all the samples, and on the right is the bacterial compositions of all the samples. Interestingly, the compositions weren't overly different. A majority of the samples had the same type of bacteria or fungus that in similar proportions to the whole. Like how all the samples have Ascomycota fungi as their most prevalent, and for bacteria, Firmicutes bacteria is the most prevalent. Seeing how most of the microbes in the samples were bacteria, this is where I tended to focus my attention. Generally, the berries all seem to result in similar compositions, but I wanted to break it down further. The most abundant sample I had access to were bananas, so I condensed all of those samples into a single chart. Pictured here are all the banana samples, and as you can see, they are all very similar. Looking past the first abundant group, the fungi in the peach color tends to be the second most abundant, and then Proteobacteria in the red and Cyanobacteria in the teal. Those tend to be in relatively equal amounts. Lastly, I compared all the stereotypical berries to one another, meaning the strawberry, blueberry, and raspberry samples. Here, the first, fifth, and sixth samples are strawberry, the second and fourth are blueberry, and the third is raspberry. We can see some variation between all the berries, but when only focusing on a specific type of berry, i.e. just the strawberries, we can see more similarities between them. Overall, this preliminary analysis revealed that berries tend to result in similar microbial compositions. So what do we do next? If I were to continue this project, I'd make sure to repeat the experiment to verify what my initial results had found. But I'd also want to compare these berries to perhaps the control samples or maybe another category of fruits to ensure that these results reflected just berries and not the entirety of the samples. Looking for a deeper meaning into these results, if confirmed, the information could be utilized in baking and knowing what fruits contribute, what microbes that could affect the chemistry of baked goods. With this information, perhaps it could help me or others discover how to make better and lighter loaves of banana bread. The possibilities are limitless, and I look forward to what else the Sourdough Project discovers in the future.